So today I'll be talking about the role of store-operated calcium channels in oral squamous cell carcinoma. And as all of you know, this is the political map of India, and this Trivandrum is here, Thiruvananthapuram in Kerala. However, if you go to the northeastern states, you can see, I don't know whether you'll be able to see my cursor. So there are 7 states, like Assam, Meghalaya, Manipur, Nagaland, Arunachal Pradesh, Tripura, Mizoram, Manipur, etc. So the IIT-Guwahati is situated near to Dispur in Assam. So this region is very important because this is one of the hotspot regions of biodiversity in the world. And the flora and fauna of this region is very unique. However, one of the main problem with this region is that it is known as the hub of cancer in India because the high incidence of cancer in this region. So this is mainly because of lifestyle and the contaminations, various metal contaminations in the soil and water, as as well as the use of tobacco and other related products. So one of the most common or prevalent cancer in this region is oral cancer. So oral cancer is the 6th most common cancer in the world, and it is the 3rd most common cancer in developing countries. So in India, oral cancer is the topmost cancer diagnosed in our country in men and 3rd among women. So in India, if the oral cancer cases are 20 cases per 100,000 population. So you can imagine the severity of this disease in our country. So what is oral cancer? Oral cancers are almost 95% are oral squamous cell carcinoma, and this is originated from the oral cavity. So either it can originate from the lip, gingiva, vulva, tonsil, floor of the mouth, soft palate, tongue, etc. And there are many risk factors of oral cancer. As I mentioned, tobacco chewing is the most common important risk factor, major risk factor of this disease. Then alcohol, and people use tobacco and alcohol together, so they will get a synergistic effect. When you use alcohol and tobacco together, the amount of the carcinogens absorbed by the cells are very, very high. Then arecanut also, chewing also is another important cause of cancer. Then different virus like HPV and then lifestyle, etc. also causes oral cancer. And there are so many genes and proto-oncogenes are involved in the development of this cancer. And as like any other cancers, development of oral cancer is also a multi-stage process. So when carcinogens attack this tongue or the epithelial cells, first leukoplakia or white patches develop, or erythroplakia, red kind of patches develop. Then dysplasia develop, then severe dysplasia, then it will turn into oral squamous cell carcinoma. So this whole process takes 15 to 20 years and also depends on the individual. So there are different therapies have been developed for the treatment of oral cancer. The most common therapy is surgery, and if the tumor is in a localized place, surgery is the most common modality of treatment used. Same, surgery sometimes followed by radiotherapy also used to kill the remnant tumors. Then if the tumor is spread to other parts of the body, then chemotherapy is the only option. However, not many chemotherapeutic agents have been developed for the treatment of oral cancer. And whatever the developed chemotherapeutic agents, this gives responds only in less than 5% of the patients. So you can imagine the problems, faced by chemotherapy. So not only that, there are many side effects associated with chemotherapy, like cardiotoxicity, neuronal toxicity, and oral mucositis, and susceptible to other different infections, etc. And uh, in addition, chemo resistance and cancer recurrence also a very huge problem in oral cancer chemotherapy. So this shows that there is an urgent need to develop novel molecular biomarkers for the prognosis and therapy for oral cancer. So, as you all know, that minerals are very, very important uh, in the body, and these minerals are very important in cell proliferation, cell division, and different biological processes and reactions inside the cells. So, one of the most important mineral is calcium, and it has been well established that calcium is very important for the growth of cancer cells, and targeting calcium machinery in a cell can be used for the prevention and treatment of cancer. So, what are the different calcium handling tools of a cell? There are many different calcium handling tools in a cell. So, it has been divided into 7 different types, like calcium ion channels, exchangers of proteins like sodium calcium exchanger, receptors, different calcium pumps, then calcium buffers, calcium sensors, and calcium sensitive cellular processors. So, these are the different calcium handling tools of a cell. Out of this, store-operated calcium channels have gained much attention in the development of different cancers and targets for developing drugs against these cancers. For example, this is an article published in January 2015. The title is Open Sesame, Pressure in Store-Operated Calcium Entry Pathway for Cancer Therapy. So, however, even though there are many calcium channels are discovered, our main focus is on store-operated calcium entry pathway. So what then? Next question is, what is store-operated calcium entry channels? So this figure is a little bit busy, but I will explain it briefly. So the calcium stored inside the cell is endoplasmic reticulum. So depends on the concentration of calcium ions in the endoplasmic reticulum. The calcium channels, open or close. So it will get more calcium if less calcium in the endoplasmic reticulum, or if more calcium is there, it will open up and then afflux calcium from the cells. So this is operated, depends on the store of calcium. These channels are operated, depends on this, concentration in the store of calcium, that is endoplasmic reticulum. That is why these channels are known as store-operated calcium entry channels. So, store-operated calcium entry channels have been reported in different cancers like breast cancer, prostate cancer, liver cancer, cervical cancer, lung cancer, etc. But oral cancer, its role has not been studied. So these calcium entry channels are controlled by different proteins like ORI1, STIM1, etc. And many studies have shown that these genes also upregulated in different cancers like glioblastoma, nasopharyngeal cancer, esophageal cancer, lung cancer, hepatoma, renal cancer, pancreatic cancer, colorectal cancer, melanoma, etc. And this table shows the different hallmarks of cancer and what are the calcium channels are involved and their expression, whether it is upregulated or downregulated, and which cancer type this has been discovered, etc. So, for example, apoptosis resistance, etc. those seen in human glioblastoma cell line and prostate cancer cells, and the proteins are involved in TRP and SOC, like ORI1. And similarly, cell migration and metastasis, these are the genes involved. Then tumor angiogenesis, these are the genes involved. And proliferation of different cancers, these are the genes involved. So therefore, we hypothesis that as oral cancers are highly proliferating cells, store-operated calcium channels may be highly active in this cancer. And if we can determine its role, we can find out the molecular signatures governing the calcium channels, and that can be used as the therapeutic targets for discovering novel molecules for the treatment of oral cancer. So our objectives of this study were the status of store-operated calcium channels in normal cells, oral cancer cell lines, and patient samples. Then the major risk factor of this cancer, tobacco, and other tobacco components on oral cancer cell proliferation, calcium signaling genes, and the store-operated calcium entry channels. Then effect of modulation, effect of SOCE or store-operated calcium entry channels in oral cancer cells using SOCE inhibitors and siRNAs. So first we checked the amount of store-operated calcium entry channels in oral cancer, and you can see if you don't give any, so this is with a multi-mode reader we have given did this experiment. So we have used one cell line or oral cancer cell lines, and if you supplied the calcium in the medium, so you can see after supplying different time intervals, if there is no calcium, in one plate in the control there is no calcium, so store operated calcium channels are not that active. However, if you supplement calcium, the intake of calcium is very high in these cells. So what we did actually, we compared the store-operated calcium entry channels and calcium intake in oral cancer cells versus normal cells. So we used a cell line called HECAT. This is a normal epithelial cell line, and SAS, this is a tongue oral squamous cell carcinoma cell line. So you can see first what we did actually with the help of some reagents, for example, tapsigargin. So that will flush out all the calcium, almost all calcium in the endoplasmic reticulum. And after flushing out, then we have provided the calcium from calcium externally to the cells. So the moment we provided the calcium, you can see in normal cells after 20-22 seconds, the amount of calcium intake was increased. However, in cancer cells, the intake was very, very, very high, almost 2.5 fold increase in calcium intake were observed in oral cancer cells than the normal cells. That shows that calcium intake is very high in oral cancer cells than the normal cells, and the calcium channels, store-operated calcium channels, may be playing some role in the development of oral cancer. So we have also determined the expression of store-operated calcium channels related genes in oral cancer cells versus normal cells. And this graph shows the fold change of the expression compared to normal cells. You can see there are 5 genes involved in this: ORI1, ORI2, ORI3, STIM1, and STIM2. So ORI1 and ORI2 were highly expressed in oral cancer cells than normal cells. And we hypothesized, and many papers also showed that these 2 genes are very, very important genes that govern these store-operated calcium entry channels. So in the next step, so this shows that in oral cancer cells, calcium store-operated calcium entry channels are very active, and the calcium store-operated calcium entry genes also highly expressed in oral cancer cells. So what about in patient samples? So we check the expression of ORI1 and ORI2 in oral cancer patient samples compared to normal tissues. You can see here ORI1 and ORI2 is less expressed in normal tissues. However, it is highly expressed in malignant tissues. And this is the graph showing the expression score of ORI1 and ORI2 in oral cancer tissues compared to normal tissues, and the expression was highly significant. In the next step, we compare the expression of these proteins in male versus females. There also we found these proteins are highly expressed in normal male versus cancerous females, male, and normal female versus cancerous females. So then, as I mentioned in the previous slides, the oral cancer can develop from any regions of the oral cavity. So we got tissues from different regions of the oral cavity, cancer tissues, and then check the expression of ORI1 and ORI2 in these tissues. And you can see in normal, we had around 10 samples, and then in cheek, the expression was high compared to normal tissue. As same results we got it in gingiva, lip, lymph node, and almost the palate and tongue, etc. This shows that calcium entry channels, or the genes or proteins that govern the calcium entry channels ORI1 and ORI2 is highly overexpressed in most of the cancers originated from different parts of the oral cavity. In one slide, I showed you that the development of oral cancer is a multi-stage process. So we examined or determined the expression of ORI1 and ORI2 in different stages of the development of oral cancer, like inflammation, benign tumor, hyperplasia, malignant tumor, metastasis, etc. In almost all cases, we have received significant upregulation of ORI1 and ORI2 proteins in oral cancer tissues and different stages of development of oral cancer compared to normal tissues. We also checked the expression of this ORI1 and ORI2 in different grades of oral cancer. There also we got significant upregulation of these two proteins, ORI1 and ORI2, in different grades like grade 1, grade 2, and grade 3 of oral cancer tissues. So, in conclusion of the first part, says that calcium entry, store-operated calcium entry channels are very active in oral cancer cells compared to normal cells. And the genes or proteins governing the store-operated calcium channels also very high in oral cancer cells. And the most important protein, ORI1 and ORI2, expression also very high in oral cancer tissues compared to normal oral tissues. So in the next step, we ask the question. So it is overexpressed. These ORI1 and ORI2 are highly overexpressed in oral cancer tissues. So what leads to the expression or overexpression of store-operated calcium channels in oral cancer? As all of you know that tobacco is the main risk factor for oral cancer. So we hypothesize that the carcinogens present in tobacco induce calcium signaling and store-operated calcium channels in oral cancer. So first what we did, we got the tobacco leaves and then prepared the tobacco extract. In our laboratory, and then we did an MTT assay, and this assay shows that if you incubate or treat the cells with the tobacco extract, you can get proliferation of oral cancer cells compared to without any treatment. And if you use the tobacco extract in the influx of calcium entry by store operated calcium channels, you can see this tobacco extract significantly upregulate the entry of calcium inside the cells. And not only that, the tobacco extract induce the expression of different genes that regulate the store operated calcium channels like RI1, RI2, TRPC1, etc. So first we check the tobacco extract. However, tobacco contains many chemicals. Excuse me. So over 3,000 chemicals have been identified from tobacco, and 60 of them are non-carcinogens. Then we checked the effect of some of the non-important carcinogens present in tobacco on store-operated calcium entry channels. So first we used NNN, that is the nitrosamine present in tobacco, and you can see that first we checked the non-toxic concentration of NNN, and then we treated oral cancer cells with the NNN, and you can see this is without any treatment. And if you treat with the NNN, the calcium entry channel uptake of calcium are very high in oral cancer cells compared to without any treatment. Not only that, RI2 expression was very high after the treatment with the NNN in oral cancer cells. Next, we tried another carcinogen, that is also another nitrosamine, known as NNK. NNK is known to cause lung cancer, and there are so many studies that shows NNK also causes oral cancer. Therefore, we checked the effect of NNK on the expression on calcium intake and the expression of different genes regulate the calcium entry channels. You can see the MTT assay shows that whatever the concentration we are using is non-toxic, and once you start treating the cells with NNK, you are getting high calcium intake by store-operated calcium channels in oral cancer cells. Not only that, as I mentioned in the previous slide, after the treatment with the NNK also, we got high expression of the genes RI1 and RI2 in these cells. Next, we used another carcinogen that is known as 4-NQO. 4-NQO is a synthetic carcinogen, and it is not generally present in tobacco. However, this is the carcinogen that we use to induce oral cancer in animals. So the advantage of this model is that if you give 4-NQO, that will mimic the development of oral cancer, human oral cancer in rats or mice with different stages. So therefore, we used 4-NQO and determine the expression of store-operated calcium channels and the expression of RI1 and RI2 and other genes. And you can see when you start treating with the 4-NQO, the intake of calcium was very high in oral cancer cells, and the genes also regulating calcium entries, store-operated calcium entry channels like RI1 and RI2 were also significantly upregulated after the treatment with the 4-NQO. So this the conclusion of this part 2 is that tobacco and tobacco components induce calcium intake and store and activate store-operated calcium channels. Not only that, the treatment of tobacco and tobacco components and the synthetic carcinogen 4-NQO induce the expression of RI1 and RI2 genes that is responsible for store-operated calcium entry channels. This shows that calcium entry channels are very important in the development of oral cancer. However, we have to again prove that with different experiments. Therefore, in the next step, we have determined the effect of store-operated calcium channel modulation in oral cancer cells using inhibitors of these channels and siRNAs. So we have used 3 different inhibitors, that is known as 2-aminoethyl diphenyl borate or 2-APB, then LSEL3 and then a SKF 96365. So these are the different inhibitors of store-operated calcium channels we have used, and we have done a preliminary study on the proliferation of effect of these inhibitors on the proliferation of oral cancer cells, and we selected the concentration that is not inhibiting the proliferation or inducing any toxicity. So then we treated this. Then we checked the effect of these channels on oral cancer cell migration after the treatment with these inhibitors. You can see that this is not treatment, this is a wound healing assay, and within 12 hours the wound is completely closed. However, when you use these different inhibitors, the wound is still remaining. That means these are inhibitors also store operated calcium entry channels, and if you use these channels, that will inhibit the migration of oral cancer cells. These results were highly encouraging, and we also studied the effect of these inhibitors on calcium intake, and we have seen that if you use these inhibitors, there is a less calcium intake by oral cancer cells. Because of the time limit, I'm not going in details. And then we used an siRNA to knock down ORI1 and ORI2. You can see that different siRNAs we used. This for ORI1 and this for ORI2. So if you use siRNA, that inhibits the expression of ORI1 and ORI2 in oral cancer cells. So then we checked, okay, so so we have with the inhibitors also, we have shown that, we have seen that if we inhibit store-operated calcium channels, there will be less calcium intake by the cells. And what about the siRNA that target ORI1, ORI2? Whether the same effect we are getting, because these are the genes that regulate the expression or activation of store-operated calcium entry channels. So we use this siRNA and determine the intake of calcium by oral cancer cells. And as shown in the figure, if you are not using any treatment, there is a high expression or intake of calcium. However, when you use siRNA, there is a less intake of calcium by oral cancer cells. siRNA for ORI1 and ORI2. That shows the ORI1 and ORI2 are highly important in activating the store-operated calcium entry channels in oral cancer cells. Then we determine the effect of this siRNA on oral cancer's migration and survival. So we have seen that if you use this siRNA or inhibit the ORI1 and ORI2, there will be less migration of oral cancer cells and less survival of oral cancer cells. This shows that store-operated calcium entry channels and its genes, that genes or proteins that regulate these channels ORI1 and ORI2 are highly important in oral cancer cell survival and migration. So we have also checked this. Yes,
Dr. Ajaikumar.
2 minutes.
I think we are running out of time.
Yeah.
So,
Sure.
I think a lot of information you have given. So please, we have to stick on the time.
In one minute, I will wind up. So we have also checked the mechanism, and then we found that this is working through the AKT-mTOR NF-kappa-B pathway. Okay. So, and not only that, there are so many genes involved in EMT and fatty acid synthase, and apoptosis. Everything was down-regulated when we inactivate ORI1 and ORI2. This is the overall conclusion. This shows that store-operated calcium entry channels are very important in the development and different process of development of oral cancer. Therefore, we can use this or target these channels to find novel drugs for the prevention and treatment of oral cancer. This is the overall conclusion and the mechanism. And we published this since last year in 2020 in the Life Science Journal. ORI1 and ORI2 regulate oral cancer cell migration and colonization. colonization by suppressing AKT, mTOR, NF-kappa signaling. And this is my group. Thank you so much.